Yep. So we're going to pick up where we left off in 19. So starting the discussion about white blood cells. So we've finished and rounded out our red blood cells. Now we want to talk a little bit about white blood cells. So these are the leukocytes. And these are normal cells. That means they have all the normal cell stuff, nucleus and stuff like that, mitochondria, not like red blood cells. One thing they don't have, however, is hemoglobin, because that's not part of their job, right? They have everything else. So what does exactly they do? Well, white blood cells, we all know, are part of our immune system. So basically they defend us against the bad guys, right? So these are the angry guys come after us, right? So that's who they go and they attack. They will also actually remove stuff. Like for instance, if there's waste, breakdown products, debris from injury, things like that, they'll actually go in there and they'll actually remove stuff. So they'll remove debris as well. From injury, toxins, for instance, if they build up in your system, and they'll remove those sorts of things as well. And then they'll also attack abnormal cells. Now, a lot of times students will be like, okay, wait a minute, what's the difference between an abnormal cell and a pathogen? Well, a pathogen, this is basically an outsider. An abnormal cell is one of the body cells gone rogue. So for instance, if there's a cell in your body that's starting to look like it's sort of jumping off the rails a little bit, your immune system will go and attack them. As a matter of fact, it does this a lot. People don't realize this, right? Because all we ever really see when it comes to things like cancer is a diagnosis, right? So we aren't really aware of it until we actually have a diagnosis. But what a lot of times people don't realize is there's cells that you have in your body that kind of start to become abnormal, and it happens actually quite frequently. Cells that could, if they left alone to their own devices, could become cancerous. And the reason why we don't get cancer is because our immune system identifies them as abnormal and then eliminates them. And there's a lot of those types of cells that get eliminated. And then the ones that kind of get through it, folks, it's not a foolproof process, right? So there are a few that sort of slip through that process, and then those are the ones that go on into cancer progression and tumor progression that we get the diagnosis from. So what we actually hear about is like the tip of the iceberg of all the things that are going on that your immune system is taking care of. And a chunk of that we're not even aware of. Like you're exposed to bacteria that you don't even know about because your immune system just takes care of the problem. Or viruses, you're constantly exposed to viruses and you don't even know it because your immune system takes care of the problem. And so there's a lot, if it's doing its job well, you never even know that your immune system is there. Okay. So that's basically what they're all doing. All your white blood cells are doing various facets of this sort of immune response. Now, if you think about it, you're probably thinking it's like, well, wait a minute, that's kind of a short list. So how come you have so many white blood cells, right? Well, think of your immune system as sort of like a military, right? The military is largely all doing the same thing, taking care of the bad guy, protecting us from harm. However, if you take a look at the military, there's a lot of complexity to the military. There's like lots of different units. There's lots of different types of soldiers, right? Some are infantry, some are more tactics. There are tank people, even just like in the Air Force, there's like pilots who do a different job than the gunner. And so there's different roles to play even within the military, but they're all needed in order to actually pull this all off. Same thing's true with white blood cells. There are different roles to play, but together they all basically have this comprehensive push to sort of eliminate the bad guy. Now, where do we find these guys? So white blood cells, we've already actually run into them, right? So if you've made it through AMP1, you've already seen a few of them. But a lot of where you see your white blood cells is in areas that are exposed to the outside world. So think about this for a second. If you get a breach in your system, a cut, like a breach in your skin, for instance, what is the first place you're going to be? Is you're in your connective tissue, right? You're immediately getting into your connective tissue. And so it makes a lot of sense that you're going to see a lot of these white blood cells hanging out there in your connective tissue, because this is likely where you're going to start to see the first entrance of these bad guys. It's a little harder because of your skin for the bad guys to get, they need access points. They can't just like cruise right on in there. They need access points. So you station your military in the most sensitive, vulnerable positions so that when the enemy does try to breach your defenses, you've got a garrison of troops ready to do battle right there. So you oftentimes see these guys patrolling around in the connective tissue all the way throughout the connective tissue. You also see this in another open system, which is part of the immune system, in the lymph organs and the lymphatic system. So the lymphatic system, which is actually part of 22, chapter 20, that's part of the take-home component of it. So it's the immune system and the lymph system. The lymphatic system is like the circulatory system, only it's an open system. So the circulatory system is a closed circuit. The immune system, or the lymphatic system, is an open circuit. So the lymph vessels are actually open-ended, like in your tissues, they're kind of like open. And what that means is that if you get a pathogen, then they can easily move into that lymph system itself, which is a good thing. Why? Because the lymph system is basically where all of your military hangs out. So for a bacteria to go into your lymph system, it's sort of like showing up, in the barracks or in the army base of your enemy. Good luck with that, right? So you're just walking right into the enemy's nest. But this is also where you see them. So this is kind of like where you house them. Your lymph nodes actually act like army barracks. Right. So an army barrack or an outpost is basically an area where you house your troops. And so it's kind of like your little camp, if you will. And if that's the enemy, that's the bad guy, it's the last place you want to be. Okay, so your lymph nodes are essentially where you're housing all of these immune cells, and then your lymph system allows you to distribute them throughout the body to be able to protect the body. We've already seen that they're small in number, right? We're talking about these things are, they're less than 1/10 of a percent. Remember, they're less than 0.1%. So overall, they're tiny, 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 tiny. If you want to put an actual number on it, I guess somebody actually decided to do this. You're talking about a few thousand per microliter. So that's like very tiny, a very tiny amount. We said that this is like having the Delta Force or the Navy SEALs in charge of your protection, not like an actual standing army. Highly trained, but not very many, right? Which makes a lot of sense because there's really two ways you can win a war, right? You can either just throw bodies at the enemy, right? If I've got more people than you, then by numbers, eventually I'm going to win. Doesn't matter if I lose most of my people, I will win. That's one way to do it. I would argue not a very good way to do it. Or they're sort of like the way of the immune system. I don't have that many troops, but they're exceptionally well trained, and my military is very well strategically organized so that I can get maximum benefit out of my soldiers. So I can beat you with tactics and strategy. And there's a long, long history of that throughout the history of war. As a matter of fact, a lot of people don't realize this, but one of the mightiest armies in history was the Roman army, right? The Romans pretty much dominated whoever they wanted for a long, long period of time. But at the apex of the Roman Empire, spread across Europe and in the Middle East and Africa, a lot of people don't realize that maintaining all of that was a relatively small army. The actual Roman army was a relatively small army, which is the reason why it blows people away that they were able to manage a good chunk of the Western world for such a long period of time. The reason why they were able to do that is because even though they were a very small army and initially a very Roman army, like you didn't join the army unless you were a true Roman, they kind of let go of that a little bit, started, recruiting other folks that led to their downfall. But the idea is they were a very, very well-trained army. So the Romans, even if you have them outnumbered, would always beat you. Why? Because they had some of the best generals and some of the best strategists and some of the best sort of infrastructural military minds. What I mean by that is knowing how to make maximum use of what you have, the soldiers you have, the training you have, the tactics you have, so that you can basically win the war. If you want to see this on display, study the Punic Wars. There's no way in the world why the Romans should have won that war, but they did. Right? The Punic Wars was basically when they took over Africa and they beat Carthage, who was kind of like their rival. And there's no reason why they should have beaten Carthage, because Carthage had the best soldiers and the best generals, but they were Romans. They were smart and clever and practical, and they just kept figuring out ways to beat you. They were very strategic in the way they did things. It's a very fascinating read, and and extended into Roman society and Roman engineering and things of that nature. It's the reason why nobody wanted to see the Romans showing up on your doorstep, because it doesn't matter where you are, they're going to figure out a way to beat you, right? And they're not going to give up until they do. Just asked how that worked with the Jewish Judea in first century Rome, when Titus destroyed the Jewish nation and basically turned Judea into what became Palestine. Right? There's a famous fortress there called Masada, which is this sort of stalwart of a thing that's up on this impossibly to get to mountain. I mean, it's like weird. It's like, it's just up here. It's like, how do you even build something up there? How did you get up there? Did you fly up there? And so it's like supposed to be this ultimate perfect fortress, right? So total protection. There's no way anybody's going to get to us, not even the Romans. Guess what they got to? So they were just that way. They were very pernicious. By the way, sounds kind of very similar to another country in a lot of ways that we know a lot about. Not quite as severe, but definitely some leanings. America is very much like that as well. So when we get kind of bullheaded about things, we are very Roman in the way we see things. We will not be denied. We will keep after you, right? Even if we're tiny. I mean, take a look at the revolution. There's no way we should have won that, but we did. So that's our Roman heritage. So we're very much neo-Romans. But anyway, that's here. That's free, by the way. That was not going to be on an exam, right? So what's the name of the fortress that the Romans took over in first century AD? For 50 points. Like, anyway. So let's take a look at this and see how we're going to be circulating these guys through here. First of all, there's a couple of things with white blood cells that are actually kind of fairly important. The first one is, unlike most cells, they can migrate into and out of the bloodstream. Cells can't do that. Most stuff in your blood can't do that. Red blood cells can't do that. The proteins that are in there, they can't do that really. I mean, so getting through the walls of your blood, and we'll take a look at what that structure is of your blood vessels in a couple of chapters. But they have the ability to do that. And the reason why they have the ability to do that is because the way they move around is by what's called amoeboid movement. Now, does anybody know what amoeboid movement is? Did you guys cover that in general bio? If you had general bio? Yeah, it's kind of like your kind of ooze, right? You sort of move around like a blob. That's kind of what you do, like kind of one of those squishies where you kind of squish it and it sort of oozes all over the place. And basically what amoeboid movement is the ability to sort of break down what's called your cytoskeleton. And this is kind of gets into a little more general biology, but the mechanism of amoeboid movement is based on the cell cytoskeleton. There's a cytoskeletal protein fiber called actin, which you guys probably remember from AMP1 fame, right, from the sliding filament model. Well, actin is also part of the overall cell skeleton to give the cell shape and structure. There are two different forms of actin in a cell's body, if you will, right? There's what's called cortical actin, which is kind of like a hard, solid type of actin that's just underneath the plasma membrane. And what this does is it basically kind of creates sort of a hard matrix to sort of maintain the shape of the actual cell membrane. So the membrane doesn't just sort of collapse down like a floppy hat, okay? And so that's kind of what that's for. So there's that what's called cortical actin. And on the inside of it, you have actin fibers that are sort of moving around in the cytoplasm that is more of a fluid actin. So it's kind of like a gel-like fluid structure with like fluid dynamics. So the way that you move around in terms of amoeboid movement is very simple. What happens is you basically, like if you wanted to move in this direction, what you do is you basically break down that cortical actin so that, I'm going to do a blow up here, so that there is a region that's now exposed with no cortical actin associated with it. Then what you do is you essentially just squeeze it and you push the fluidy actin against that membrane, and it's just going to bulge out just like a water balloon, just like squeezing a water balloon. And then you basically can just sort of attach that to wherever you are, and then you can maintain that by rebuilding your cortical actin and giving yourself that structure and that shape once you get into that area. So the way it oozes is with this sort of reconstruction of its cytoskeleton. Amoeba do that. So they're kind of like oozing around like a blob, but our white blood cells do the same thing. And the reason why they can migrate through the blood vessels is because think of the blood vessels sort of like a picket fence, right? A picket fence has gaps in between each of the pickets. One of the things the white blood cells can do is they can be attracted by different chemical stimuli. So we have what's called positive chemotaxis. Now, what does that mean? So chemotaxis basically means, so chemo is associated with smell, and to be tactic is to walk. So this is kind of like smell walking. That's kind of what that is. And here's what they do. So you have some sort of a chemical stimulus, right? And if it's positive, that means it's attractive to you. If it's negative, that means it's repulsive to you, okay? So if you have positive chemotaxis, what happens is the white blood cell moves toward it. Very similar to like if I'm in my backyard and I'm a white blood cell and I smell my neighbor barbecuing. For me, because I'm not a vegan, that is an attractive thing. Now, if you're a vegan, that's probably a negative thing, right? So you're getting away from it. But for me, it's a positive thing. So I can smell it. And then what do I start doing? I start to move toward the smell. That's exactly what white blood cells do. They can smell these chemicals and they basically move toward it. Now, here's the problem. I've got a picket fence between me and my neighbor. Is that going to stop me? No, because I move by amoeboid movement. What I do is I basically just sort of ooze myself in between the gaps of the picket. I just kind of like putty, just sort of squeeze in between there. And then here I am eating your hamburgers. Right. So I just invited myself to the party. Right. That's kind of what I did. But that's essentially exactly what white blood cells do. They're in the bloodstream, and then there is a positive signal, some sort of a chemical signal, oftentimes either emitted by other white blood cells during battle or by toxins being emitted by bacteria. But either way, there's some sort of an attractant, and the white blood cells are able to ooze between the gaps, and those are not very big gaps, they're very tiny, but they can ooze between those gaps, and they can basically pop out into the tissue, to the connective tissue, where they can start to do battle with the bad guys. So that's basically how they work. They're the only cells that can really do that, and that's kind of what they're doing actually throughout the body. They're just sort of oozing all over the body, sort of tumbling around, patrolling different areas, and there's nothing really that can keep them from moving through those fences. So if I wanted to basically start oozing through the fences of all my entire neighborhood, I totally could. right? Just kind of like oozing all the way through there. It kind of reminds me of a short story that I read once in college. I forget who did it, but it's called The Swimmer. It was a fascinating little thing. It was a guy who basically got off work and he wanted to swim. And what he ended up doing was some crazy thing. Between his work and his house, he basically went from one yard to the next and he swam through people's pools. So it's like he would jump the fence, he would start on one end of the pool, he would swim to the other end of the pool, and then he would jump the fence, and then he would jump into the other person's pool, and then he would swim. And it was like, it was hysterical. But I knew what he was talking about because it was like based in LA. And it's like, yeah, you could do that in LA. You could do that in LA. Because it's like one pool after another. So, and it was just funny. It was actually a pretty good story. Totally useless, but could be that useless. I didn't remember it. So it did its job. Now, some of these guys are phagocytic. What does it mean to be phagocytic? So what's phago mean? Phago means to eat. Cyte is the root for cell. So this is a cell eater. So phagocytic cells engulf other cells and devour them. That's what they do for a living. There's several different phagocytes in your immune system: neutrophils, eosinophils, and monocytes. Those are all phagocytic cells. So they will actually kill cells by engulfing them, basically gobbling them up. Okay, which is kind of cool. Okay, what kinds of white blood cells do we have? Well, there's our list. We got neutrophils, eosinophils, basophils, monocytes, and lymphocytes. That's the list. So we're going to basically talk a little bit about these, and we're going to sort of break these down type by type and kind of get a sense of sort of what they're doing. So here's basically what they look like histologically. You can see this is like really, really nice images, obviously. There's two major subgroups that are associated with this. The first group here is what's called granulocytes. And the reason why is because they have these cytoplasmic granules. You'll see this kind of like little grainy texture that's associated with all of them. And in those granules are actually necessary proteins and things like that, molecules that they use for signaling and communicating with other immune system. The reason why the immune system is really it's really helpful to think of it as a military is because when you study how a military operation works, the one thing that'll blow you away is how good the communication is in the military. The fact that the communication is really, really good. So all of the officers are communicating with each other. They're getting messages to the battlefield, and it's designed to be a very, very strong, integrated, communicative situation, so that everybody knows what they're supposed to be doing, and it's pervasive throughout. So that's what those granules are for. It's for communication with the rest of the troops, so that you can organize your attack. Now, the other two are agranulocytes. They do not have cytoplasmic granules. Okay. And so this is a pretty good representation. So here you can see your neutrophils. You can identify them because they've got these little puckered lobe-like nucleus. Notice there's deep constrictions in there. That's very neutrophilish. And generally speaking, it kind of stains in a right stain, kind of on the bluish side. So a lot of you guys have seen this already in your blood smears, right? So these are pretty easy to identify. The eosinophils are a little bit different. They have a similar sort of a nucleus. It's still puckered, but the granules stain bright red in a right stain. And so these guys histologically have similarity to neutrophils, but they stain differently because they've got different things in those cytoplasmic granules that are for their communication. The basophils are a little bit different. You can still see that you got that very strong sort of puckering effect, right? That poodle balloon effect on the nucleus, but the granules tend to stain usually dark blue, kind of a navy blue sometimes. If you have a cell where you can sort of see a little hint of a poodle balloon nucleus in there, but it's hard to see because the granules are so dark, they're obscuring it, then you're probably looking at a basophil. It comes across as very kind of a navy bluish sort of a look. Those are your granulocytes. Now the agranulocytes, basically, there's really two major types. The monocyte, which is basically you notice the relative clarity of the cytoplasm, and I do say relative, right? So it's not like it's absolutely clear. There is still stuff in the cytoplasm. It's just not as dominant a feature as you see, for instance, in a neutrophil. The other thing also that's a giveaway is the horseshoe-shaped nucleus that has no puckering, right? So basically, you don't see that extreme sort of constriction that you see in the granulocytes. So that's the giveaway of the monocyte, is a nucleus that seems to be in varying shapes. So it could be a total horseshoe, a comma, but it's got this sort of continuous thickness throughout the nucleus. And then of course the lymphocyte, which has a nucleus that mostly is filled up the cell. So it's mostly full. And especially in ours, we don't even hardly see the halo of cytoplasm around our lymphocytes. It's so full. Okay. But if you do see it, you'll notice it's going to be a clear, it'll be a kind of a clear cytoplasm. So these are the big E's, right? These are your 5. Now the question is, what do they do, right? So we got that far so far, right? We got that far in lab. So what's new here? Well, let's take a look at what's new. Let's take a look at actually what they do, and then we'll take a look at how we make them, right? So those are the pieces that we're going to take a look at with our white blood cells. So the neutrophils are the first one we're going to take a look at. Now the neutrophils have a really fancy dancy name to them, right? Polymorphonuclear leukocytes. That's if you want to impress your friends, right? Use that one on them. You go to a party, lay that one on them. You'll never be invited again, but you'll sound really smart, right? So those are basically neutrophils. These, by the way, are the dominant representative of your white blood cells. These are the most abundant. So they make up between half to almost 3/4 of your total circulating white blood cells. The reason why they're important is because these guys are also the first responders. So whenever you get infected with a bacterium, the first one to respond, the first one on the scene is going to be the neutrophil. Okay. They're also phagocytic. Which means they gobble up the bad guys. But here's the problem. A neutrophil, when it kills, it basically kills like a bee. How does a bee kill? It stings you, right? And then what happens? Like a honeybee, right? So if you've ever seen one, it kind of like crawls off, right? And what happens to it? Yeah, it dies, right? If you've actually seen it, what actually happens is the actual tip of the abdomen actually rips off of the bee. So it's like part of the bee's body as actually ripping off it as it's like pulling away. Why? Because what's inside the tip right there, while the stinger, is the poison sac. And so when it stings you, it basically walks off, which kind of rips off part of its abdomen. Obviously, if you're ripping off part of your body, you're probably not going to be around for much longer, right? Which is the reason why the bee stings and then it dies, because it basically rips part of its abdomen off in the process of stinging you. And if you actually see what's left behind, you see that little chunk of abdomen on there, right? But you can see that little poison gland on the stinger, and it's still pumping. So it's like he's still like going away at you, right? So that's pretty wicked. So it's like, you know, it's pretty bad. So a bee basically kills once and dies. So what happens with a neutrophil? It kills once, but it dies in the process of killing. So you're constantly having to replace them, but also in a battlefield, you're going to see an accumulation of dead neutrophil bodies. When that happens, they accumulate and they start to create a whitish appearance that we know as pus. So pus is basically the accumulation of neutrophils that have died. The bodies have piled up. That's the reason why they're called white blood cells, because pus is white, right? So they died. Their little bodies are piling up in the process of doing battle, and they're just a big pile of neutrophil bodies. It's basically what pus is. But that's the reason why when you see pus or a wound that has a lot of pus in it, it's a bad sign. Because what that means is you've got a wound that's infected. The more pus you have, it means the more strenuous that invasion was. So the bad guys are really on you because you've got all your immune system is just like sacrificing tons of neutrophils in trying to get this thing under control. Right? That's the reason why having a pussy wound is a bad sign, is because it means that the attack is pretty severe. And not only that, but it also means that it's not over, right? Because the rest of the immune system has to kind of get in there and sort of back them up. Okay. So these guys are granulocytes. So what's in those granules? Well, for these neutrophils, lysosomal enzymes, these are digestive enzymes. That will degrade the biological molecules like protein, DNA, things like that, carbohydrates, the stuff that the pathogens are made out of, right? So these are digestive enzymes. So when you degranulate, then you're basically sicking these digestive enzymes on this pathogen, and you're basically breaking them up. There are also going to be some bacteriocytes. So these are molecules that actually act like as antibacterials or inhibitors to the bacteria itself. So for instance, oxidative stuff like hydrogen peroxide, superoxides, pretty nasty stuff. These will kill a lot of bacteria because bacteria aren't used to dealing with oxygen. So they don't have the defenses that we do against these sorts of things. Okay. And so you can kill them with these compounds. It's sort of like trying to kill us by unleashing a bunch of cyanide on us. Right, that's a good way to kill us because we don't have a mechanism to deal with cyanide poisoning, right? Just like the bacteria don't have a mechanism to deal with these oxidative products, these like peroxide and superoxide and things like that. So that went a lot longer. I got a little too excited there. I went way longer than I intended to. I apologize. I just glanced up at the clock, so I want to stop there. Because I was only intending to go for 20 minutes, but we'll talk about neutrophil action and continue our list later on. So I want you guys to be able to dig into your lab.